Microsporogenesis. Microsporogenesis means microspores are developed from the microsporangium. Microspores are developed from microsporangium is called microsporogenesis. The microsporangiums are present in anthers. Microsporangiums are present in anthers. Dithecous anther containing 2 microsporangium chambers in one lobe. Another 2 microsporangium chambers present in one dithecous. Means dithecous anthers contains 4 microsporangium chambers. Monothecous anthers contain single anther lobe. In single anther lobe, 2 microsporangium chambers are present. 2 microsporangium chambers are present in monothecous. In dithecous, 4 microsporangium chambers are present. From that microsporangium chambers, microspores means pollen grains are developed is called microsporogenesis. Development of microspores or pollen grain is called microsporogenesis. See, here it is microsporogenesis. It is the dithecous anther TS, transverse section of dithecous anther. The dithecous anther containing 2 lobes. It is one lobe, it is another lobe. In one lobe, 2 microsporangium chambers. Here it is one microsporangium chamber. Here it is another microsporangium chamber. Total in dithecous anther, 2 lobes are present. In each lobe, 2 microsporangium chambers are present. Total 4 microsporangium chambers are present in dithecous anthers. From these microsporangium chambers, pollen grains are developed, called microsporogenesis. See, young age of anther, dithecous anther transverse section shows 4 microsporangium chambers. 1 microsporangium chamber, second one microsporangium chamber, third one microsporangium chamber, fourth one microsporangium chamber. 4 microsporangium chambers are present in dithecous anther. We observed each microsporangium chamber at the matured stage. Some important walls are present. See, children, it is the microsporangium chambers. Here also microsporangium chamber, here also microsporangium chamber. We observed one microsporangium chamber enlargement. It shows epidermis, means each microsporangium chamber covered with epidermis. It is a single layer. It protected to inner parts and it gives a perfect shape to microsporangium chamber. It made with rectangular parenchyma cells. Below the endodermis, endothecium is present. Generally, endothecium cells are large in size. They are contain large nucleus. They are large in size. They are rectangular in structure or pentagonal or hexagonal in structure. It present beneath of the epidermis. Beneath of the epidermis layer is called endothecium. Below the endothecium, 2 layers of parenchymatous cells are present. The 2 layers of parenchymatous cells are called middle layers. The middle layers are present beneath of the endothecium layer. Below the middle layers or beneath of the middle layers, tapetum layer is present. The tapetum layers are large in size. They are hexagonal or polygonal or rectangular in size. Each tapetum cell containing one large nucleus. The main function of tapetum cells, supplying of food material to microspore mother cells. Below the tapetum cells, diploid cells are present. The diploid cells are called microspore mother cells. The microspore mother cells need nutrients. The nutrients collected from tapetum cells. Nutrients collected from tapetum cells. Mainly in angiosperms, the tapetum cells are two types. One is glandular tapetum. Second one is amoeboid tapetum. See in this diagram, glandular tapetum cells are present. Glandular tapetum cells are containing perfect cell wall around the tapetum cells. See in this diagram, perfect cell wall is present. If around the tapetum cell, perfect cell wall is present, that tapetum cells are called glandular tapetum cells. Glandular tapetum cells. Another one tapetum is amoeboid tapetum. Amoeboid tapetum cells are without cell wall. Around the tapetum cells, cell wall is absent. The tapetum cells are called amoeboid tapetum. That amoeboid tapetum containing only periplasmodium layer around the tapetum cells. Whatever it may be, the glandular tapetum cells or amoeboid tapetum cells, both tapetum cells are contains nutrients. That nutrients are supplies to microspore mother cells. From the endothecium to tapetum layer, endothecium to tapetum layer means endothecium, middle layers, tapetum. These three types of layer cells are called anther wall. Three types of layers of cells are called anther wall. Outer layer of anther wall is epidermis. Below the epidermis, endothecium. Below the endothecium, two middle layers. Below the two middle layers, tapetum layer is present. In this anther wall, major important layer is tapetum. The tapetum cells are contains high amount of nutrients. That high amount of nutrients are supplies to microspore mother cells. In angiosperms, tapetum cells are mainly two types. One is glandular tapetum. Glandular tapetum cells are containing perfect structure. Each glandular tapetum cell containing cell wall. And second tapetum is amoeboid tapetum. They didn't have any proper structure. They look like a amoeba. Amoeba without perfect structure. Similar amoeboid tapetum cells also present in few types of angiosperms. The both type of tapetum cells, they are glandular tapetum cells and amoeboid tapetum cells. Supplies nutrients to microspore mother cells. See, below the tapetum layer, more number of diploid cells are present. The diploid cells are called sporogenous tissue or microspore mother cells. Sporogenous tissue or microspore mother cells. Microspore mother cells are diploid in condition. The diploid condition of microspore mother cells participated in meiotic or reduction division to form 4 haploid spores. The 4 haploid spores are called pollen grains. 4 haploid spores are called pollen grains. See children, it is sporogenous cells. The sporogenous cells are also called microspore mother cells. The microspore mother cells are in diploid condition. The diploid condition microspore mother cells are participated in reduction or meiotic division to form 4 haploid spores. The 4 haploid spores are called microspores. The microspores are also called pollen grains. Microspores are also called pollen grains. Here it is, microsporangium chamber produce microspores is called microsporogenesis. Microsporogenesis, microspores means pollen grains are developed from microsporangium chamber is called microsporogenesis. See, diploid microspore mother cells participated in the reduction or meiotic division in 2 ways. One is successive type of meiotic division. Second one is simultaneous type of meiotic division. First we discussed about the successive type of meiotic or reduction division. Diploid microspore mother cell participated in first meiotic division. See, meiotic division containing meiotic 1 and meiotic 2 divisions. Microspore mother cells containing diploid nucleus. The diploid nucleus of microspore mother cells participated in first meiotic division to form 2 diad cells. The 2 diad cells are developed by the first meiotic division. See, it is 2 diad cells. Each diad cells containing diploid nucleus. Around these diad cells, perfect cell wall is present. Mean, in meiotic division 1, karyokinetic divisions are completed. Karyokinetic divisions means nuclear divisions. After karyokinesis, 2 diploid nuclei are developed. After karyokinesis, cytokinesis division will occur. By the cytokinesis division, cytoplasm divided into 2 half parts. Finally, between these 2 nuclei, new cell wall is formed to form 2 diad cells. Means 2 clear structure of diad cells are formed in successive type of meiotic division. The 2 diad cells are again participated in meiotic 2 division. The 2 diad cells are again participated in meiotic 2 division. In meiotic 2 division, the 2 diploid nuclei are participated in karyokinesis after cytokinesis. After meiotic 2 division, 4 haploid nuclei are formed. After karyokinesis, cytokinesis division will occur. After cytokinesis division, cytoplasm are divided into 4 equal parts. Finally, around the haploid nuclei with cytoplasm, cell wall is formed to form 4 haploid microspores. That microspores are also called pollen grains. This type of microspore mother cell division is called successive type of meiotic division. Successive type means microspore mother cell participated in first meiotic division to form 2 diad cells. After the first meiotic division, the two dyad cells are participated in second meiotic division. In second meiotic division to form 4 haploid microspores. The 4 haploid microspores are also called pollen grains. Another type of microspore mother cell division, meiotic division, is called simultaneous type of meiotic division. Simultaneous type of meiotic division. In this simultaneous meiotic division, first microspore mother cell containing diploid nucleus. The diploid nucleus participated in only karyokinesis divisions. That karyokinesis divisions are meiotic 1 and meiotic 2. First meiotic division 1, in karyokinesis 2 nuclei are formed in the mother cell. The 2 nuclei are again participated in second meiotic division of karyokinesis. Finally, they develop into 4 haploid nuclei. After 4 haploid nuclei, cytoplasm is divided into parts, 4 parts. See, in this simultaneous, 2 nuclei are formed by the first meiotic division. The 2 nuclei again participated in the second meiotic division to form 4 haploid nuclei. The 4 haploid nuclei are completed by second meiotic division. After karyokinesis, cytokinesis divisions are occurs. After cytokinesis division, 4 haploid microspores are formed. See, it is the major divisions of the microspore mother cells. First division is successive type of meiotic division. Second one is simultaneous type of meiotic division. Simultaneous means first divisions are karyokinesis divisions. First divisions are karyokinesis division. Karyokinesis division 1, karyokinesis division 2. Then after cytokinesis, karyokinesis division simultaneous with cytokinesis divisions to form 4 haploid microspores. The 4 haploid microspores are also called pollen grains. In successive division, first meiotic division is completed to form 2 cells. The 2 cells are again participated in the second meiotic division to form 4 cells. The 4 cells are in haploid condition. We call successive type of meiotic division. In simultaneous, first divisions only karyokinesis divisions to form nuclei. 2 nuclei. The 2 nuclei again participated in the karyokinesis. They form 4 haploid nuclei. The 4 haploid nuclei are present in only microspore mother cell. Finally, after second karyokinesis division, cytoplasm divisions are occurs. The cytoplasm divisions are completed to form 4 haploid nuclei. See, the both divisions occurs in the microspore mother cells. One is successive type of division, second one is simultaneous type of division. By these divisions, 4 haploid microspores are formed. The 4 haploid microspores are arranged in tetrahedral position. Means after completion of meiotic division, the 4 pollen grains or 4 microspores are fused at one place. They cannot leave from each other. They are fused. That position is called tetrad. Tetrad position means 4 pollen grains are arranged in a ball-like structure, in spheroidal structure. Here it is 3 pollen grains are visible from this direction. Another pollen grain also presented outside or back side of these 3 pollen grains. Total 4 pollen grains are present in a ball or in a spheroidal-like structure. This arrangement of pollen grains is called tetrahedral microspores. Tetrahedral microspores. And second type one is isobilateral. See, 4 pollen grains or 4 microspores are formed at end of the meiotic division. From these 4 microspores, 2 microspores are present at upper tier and 2 microspores are present at the lower tier. They are similar in structure. This arrangement of microspores is called isobilateral arrangement. Another one is decussate arrangement. 4 pollen grains or 4 microspores are arranged in a 2 tiers. Upper 2 tier microspores are opposite lower 2 tiers of pollen grains or microspores. See, here it is. Upper 2 tier pollen grains are visible and lower tier 2 pollen grains are arranged in the opposite manner. In this also 4 microspores are present. This arrangement is called decussate arrangement. And the next one, microspore arrangement, is called T-shaped arrangement. In T-shaped arrangement, 2 microspores are present in upper tier and remaining 2 microspores are present one above the other. They are look like a T shape, capital T in shape. This arrangement is called tetra T-shape arrangement of microspores. And the last one is linear microspores, linear arrangement of microspores. See, in this linear arrangement, 4 microspores are arranged in a line, one above the other, one above the other. They look like a tire-like structure. In each tire, 1 microspore or 1 pollen grain is present. These are the types of microspores, different types of microspore tetrads. One is tetrahedral, second one is isobilateral, third one is decussate, fourth one T-shaped tetrahedral tetrads, and fifth one linear arrangement. Finally, microspores mother cells are participated in the meiotic division. They produce 4 pollen grains. They are in haploid condition. The 4 pollen grains are also called microspores. When the matured stage of microsporangium chamber, all microspore mother cells are participated in the meiotic or reduction division to produce more number of haploid pollen grains. At this time, on this anther, dithecous anthers, notch-like structures are present. The notch-like structures are containing special types of tissues. The special types of tissues are called stomium tissues or stromium tissues. The stomium tissues or stomium tissues are in hygroscopic condition. The hygroscopic condition stromium tissues or stomium tissues loses their water at the mature condition and they are ruptured. The ruptured area of stomium tissues, microspore or comes out from the microsporangium chambers. See, in one lobe between 2 microsporangium chambers, endothecium layers are disappeared or degenerated, middle layers are disappeared or degenerated, tapetum layers are disappeared or degenerated. Finally, at the matured stage, in each microsporangium chamber, only haploid pollen grains are present. That haploid pollen grains comes out from the stomium tissues. See, stomium tissues are ruptured. From this area, haploid microspores comes out from the microsporangium chamber. This development is called microsporogenesis. And at early stage, the 4 microsporangium chambers need food material. They are received by connective tissues it containing xylem and phloem. From this chambers, microsporangium chambers. Haploid microspores are comes out from, and they are light in weight. They are moved by wind or moved by insects, moved by the birds, moved by the snakes, moved by the different types of living organisms like bats. That process is called pollination mechanism. Now up to here in this topic, microsporogenesis is completed. Microspores are developed from the microsporangium chamber is called microsporogenesis.